Hello guys, this is Dr. Naveen YP. I have done my PhD in biochemistry and I am currently teaching biochemistry to the post-graduation students of Adi School of Natural Sciences, Adi University. Now in this lecture, we will discuss on the glycolysis, its history and its importance. Now, before going to uh, actual understanding the pathway through which the pathway of this glycolysis, we have to understand what is its importance. Then we can easily understand what is that particular pathway. Now we know that we require energy. I mean, all living organisms require energy, and the ability to consume energy and convert energy and store energy is what makes uh, living organisms unique than the non-living organisms. Now, as I stated, we continuously require energy for all the process that we do. For instance, we require energy to move, to sense, like to see, to taste, to hear, to touch, to think, to respire, to reproduce, to excrete, every conscious and unconscious things that are happening inside our body, we continuously require energy. Now, without this energy, we cannot survive. We continuously require energy. Now, how do we get the energy? Now, we consume that energy in the form of food. And that food consists of carbohydrates, lipids, and proteins. Now, these are the three important biomolecules that are very crucial to sustain life. Now, carbohydrates are the major source. We require about 120 gram of carbohydrates per day. Now, about 98% of the carbohydrate that we consume either contain glucose or it will be converted to form glucose. Now, what does this statement mean is that now we consume carbohydrate majorly in the form of glucose. Now, glucose consumed, whatever the glucose that we consume will yield some amount of energy, and that energy we can utilize for all our requirement. Now, how this glucose will yield some amount of energy, I mean yield energy, is the topic of this particular lecture. Now, if you see here, it is a molecular mechanism from which glucose that we consume undergoes certain chemical changes to yield energy. Now, in metabolism, we have catabolic pathway and then we have anabolic pathway. The catabolic pathways are the ones which yield energy, and that energy is utilized by our body for its sustenance of life. Now, if you see here, now this glycolysis is a very ancient mechanism. That what does that mean? It means that it is originated in the microorganisms that were living 3.5 billion years ago. Okay, that is it is one of the oldest pathway that exists, and this particular pathway is the one which exists in E. coli and even in humans. And this particular pathway, glycolysis, is responsible for liberation of energy, that can be utilized by any kind of organisms for the sustenance, I mean survival and sustenance of molecular growth. Now, when it comes to this pathway, though it existed since 3.5 billion years, and this pathway was reduced recently, I mean when compared to that 3.5 billion years, recently in 1950. Now, it took about 80 years to completely reduce this pathway. That is this glycolysis, or a chemical pathway. It is also called EMP pathway, which stands for its discoverers Embeden, Meyerhoff, and Paranas. These are the three German scientists who actually worked to understand how this glucose will be converted into pyruvate. Now, if I have to define biochemically, glycolysis is a metabolic pathway that converts a molecule of glucose into two molecules of pyruvate. Now it happens inside the cytosol of all the cells. Now during the conversion of glucose to pyruvate, there will be liberation of some amount of free energy, and that free energy will be utilized to synthesize ATP and NADH. Now these ATP and NADH will be utilized for all other biochemical reactions that require energy. Now, if I have to give an overview before we completely go to the mechanism and the chemical reactions, stepwise chemical reactions. Now, glycolysis is a 10-step chemical pathway, biochemical pathway, which happens in the cytosol, and it has 10 enzyme steps, and it yields two ATP molecules and two NADH along with two pyruvate molecules. Now, overall reaction of this glycolytic pathway can be given in this balanced chemical equation, where you can see glucose in the presence of 2 NAD, 2 ADP and 2 PI with the aid of 10 enzymes will get converted into two molecules of pyruvate, two molecules of NADH plus 2H plus and two molecules of ATP and two molecules of water. Now, the first reaction is that conversion of glucose into glucose 6-phosphate. Now, this particular reaction is catalyzed by an enzyme called hexokinase. Kinases are the enzymes. These are responsible for transfer of a phosphate group from ATP to the target molecules. In this case, hexokinase, what it will do is that it will take a phosphate group. And it adds that phosphate group to carbon number 6 of the glucose. Now, if you see the structure of glucose, this is the Haworth ring structure of glucose. Now, this is 6-membered ring, which is formed by 5 carbon atoms and 1 oxygen atom, you can see. And on each carbon, we have H and OH. And this is alpha D glucose, because if you see here, the OH group is projecting downwards. And then this is carbon number 2, where it contains H and OH. This is carbon number 3. Again, it contains OH and H. And then this is carbon number 4, which contains H and OH. Next one is carbon number 5. It contains CH2OH and H. Now, this is the structure of glucose, and this structure is easy to remember. If you remember this particular structure, then it is very easy to follow up the subsequent reactions, subsequent chemical reactions. Now, the first reaction, as I told you, this is a reaction where the glucose is primed to become to undergo further chemical alterations to yield energy. Now, this first reaction is catalyzed by an enzyme called hexokinase. Hexokinase is an enzyme which takes this phosphate group. If you see here, this is ATP, the structure of ATP. Now, in the structure of ATP, we have different components. You can see here, this is the nitrogen base or adenine base. This is the ribose, and these are the 3 phosphate groups. And these 3 phosphate groups are named as, this one is alpha, this is beta, and then this one is gamma phosphate. Now, what this hexokinase will do is that it takes this phosphate group from the ATP, this molecule. Now, ATP is a coenzyme here. Now, hexokinase binds both to glucose and ATP, and from the ATP it takes this gamma phosphate group and it transferred that phosphate to 6th carbon of the glucose molecule. This is what happens in the first reaction, where glucose will be converted into glucose 6-phosphate. Now, all the cells in our body, they have glucose transporters. Now, these glucose transporters are responsible for taking up the glucose from the extracellular fluid and putting that into the cytosol. Now, once it comes to the cytosol, there is a high chance that the glucose molecule can go out through that same glucose transporter. Now, when this glucose is converted into glucose 6-phosphate by this enzyme called hexokinase, now that phosphorylated glucose is blocked. I mean, it is blocked inside the cell. Now it cannot go out, cannot go out of the cell. Now, if you see the structure, this is the basic structure of glucose and this is the structure of glucose 6-phosphate. There is nothing changed here. Only thing is that there is an addition of, instead of this hydrogen, okay, we have added here, there is an addition of the phosphate group to form glucose 6-phosphate. Now, if you, the overall reaction I have written here, what happens is that glucose in the presence of ATP, it will get converted into glucose 6-phosphate. 2 minus, there is a charge on the glucose 6-phosphate. If you can see here, there is O minus 1, O minus 2. These are the 2 minus charges that are here. So that is why we write glucose 6-phosphate 2 minus plus ADP 3 minus. Now, if this phosphate is gone, if this particular phosphate is gone, again there will be a negative charge over here. Because of that, there is an 1, 2 and 3 negative charges. That is what written here, ADP 3 minus plus H plus. Now, this is a transfer reaction where the phosphate group is transferred from ATP to glucose to form glucose 6-phosphate. Now, this reaction is highly irreversible because the delta G for this reaction is minus 34 kilojoule per mole. Because of the, now what does that mean? When the glucose gets converted into glucose 6-phosphate, okay, glucose 6-phosphate, there will be liberation of minus 34 kilojoule of kilojoule per mole of energy. Because of that energy, there is a high energy difference between the glucose and glucose 6-phosphate. Because of that, this reaction is highly irreversible. This is second step of glycolysis, where glucose 6-phosphate will get converted into fructose 6-phosphate. Now, if you see here, glucose 6-phosphate and fructose 6-phosphate, they are isomers. These are called functional isomers. Glucose and fructose are functional isomers. The only difference that they have is that the glucose bears an aldehydic functional group, whereas the fructose bear ketone as a functional group. Now, in this particular step, glucose 6-phosphate will get converted into fructose 6-phosphate. And this particular reaction is catalyzed by an enzyme called phosphoglucose isomerase. The enzyme is given the name, the enzyme has been given the name isomerase because this enzyme isomerases aldehyde form of this glucose into ketose form, which is fructose 6-phosphate. Now, this particular step is very necessary because now when this fructose 6-phosphate is there in the down the steps, fructose 6-phosphate will get converted into fructose 1,6-bisphosphate, and then it can properly cleaved in equal halves to generate 2,3 carbon sugars. So, and also if you this fructose can form carbanion stabilization. That is very important during the breakage of this 6-carbon compound into 2-carbon compounds. Now, this reaction is highly reversible because now if you have if you compare here, the delta G is minus 2.9 kilojoule per mole. This is very there is no much difference between the energies of the reactants and products. When there is less difference of energy between the reactants and products, the reaction will become freely reversible. Now, this is the overview of the reaction. We see here glucose 6-phosphate getting converted into fructose 6-phosphate. The type of chemical reaction is isomerase. Now, the actual thing that happens here is that aldose, the glucose, get converted into a ketose, which is fructose, and enzyme, this particular reaction is reversible. This is third important reaction of glycolysis, where uh, fructose 6-phosphate will get converted into fructose 1,6-bisphosphate. The enzyme which catalyzes this particular reaction is phosphofructokinase. Uh, if you see here, there is no much difference between fructose 6-phosphate and fructose 1,6-bisphosphate. Now, in case of fructose 6-phosphate, there is a phosphate group. This is the phosphate group which is attached to carbon number 6 of this particular fructose molecule. Now, this molecule will be added with one more phosphate group by enzyme phosphofructokinase. Now, kinases are the enzymes I had told you. These are the enzymes which transfer the phosphate group. What this phosphofructokinase will do is that it takes a phosphate group from the ATP or gamma phosphate group, terminal phosphate group of this ATP, and then transfer that onto carbon number 1 of fructose 6-phosphate. Now, when the phosphate group is gone, ATP will get converted into ADP. Now, this molecule, it will become fructose 1,6-bisphosphate. Fructose 1,6-bisphosphate. Now, if you see the structure, this is called furanose structure. Furanose structure is nothing but the molecules which are formed by 5 carbon rings, or the molecules which contain a ring structure formed by 5 uh, atoms or groups. Now, if you see here, there is an oxygen here, there is a carbon here, there is a carbon here, there is a carbon here, and then there is a carbon here. This structure is called furanose structure. Now, in this furanose structure, we have the CH2OH, OH, and then CH2OH, H. Now, this is carbon number 1, this is carbon number 2, carbon number 3, carbon number 4, and then carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5, and then carbon number 6. There are 6 carbons in the fructose. Now, this fructose 6-phosphate will get converted into fructose 1,6-bisphosphate. If you see here, this out of this molecule, okay, all other things remain same in this particular reaction. Only thing that happens here, instead of the hydrogen, there will be addition of a phosphate group to form fructose 1,6-bisphosphate. This reaction is highly exergonic in nature. Exergonic, that does mean that it has high delta G value is highly negative. What does that mean? When the fructose 6-phosphate will get converted into fructose 1,6-bisphosphate, it liberates too much. It will liberate too much of amount of energy, which is 90 kilojoule per mole. Because of that, this particular reaction is highly reversible. And this phosphofructokinase is also one of the important regulatory enzyme of this glycolysis, and it is regulated by various other components. Okay, so those regulatory components will help in fine tuning the flux or movement of, or the speed of operation of this glycolysis. So this is overall reaction here that I have written. Fructose 6-phosphate 2 minus, because if you see here, this is the structure of fructose and this is the structure of phosphate which is attached to the carbon number 6, or you can say the O and H of this fructose is being replaced by this phosphate. 2 minus is because there are 2 oxygen atoms in the phosphate which carries two negative charges, negative charge 1, negative charge 2. Because of that, I have written, it is written here fructose 6-phosphate 2 minus. Now ATP 4 minus, because in ATP there are 4 oxygen atoms which carries negative charge, and then that will become fructose 1,6-bisphosphate, again 4 minus here, 4 minus, because if you see here, there is a negative charge here 1, negative charge 2, negative charge 3, and then another oxygen will be there here which carries negative charge 4. Because of that, we have written it fructose 6-bisphosphate 4 minus. When ATP is getting converted into ADP, now there is one phosphate ground. If one phosphate goes out, there will be one negative charge less here, and then there will be a production of proton. Now this is a type of chemical reaction called transferase chemical reaction. Transferases are transferase means a group is being transferred from one molecule to another. Here in this context, a phosphate group is being transferred from fructose from ATP to fructose 6-phosphate to make it fructose 1,6-bisphosphate, and this is a committed step. Once this particular step happens, then there is no other chance that it can go come back, and then this reaction is highly reversible. Now, why it is called committed step? Now, if you see here glucose 6-phosphate, this glucose 6-phosphate can enter other pathways also. Glucose 6-phosphate can get converted into, can enter into the synthesis of glycogen. Glucose 6-phosphate can enter into HM pathway, or glucose 6-phosphate even can into glucuronate pathway. Now, once if it converted into fructose 1,6-bisphosphate, there is no other uh, option for this particular molecule. It has to come down through this particular steps to get converted into pyruvate. That is why it is called a highly committed step of glycolytic pathway. Dihydroxyestone phosphate and the glyceraldehyde 3-phosphate are formed in this reaction. Now, glyceraldehyde 3-phosphate, this glyceraldehyde 3-phosphate which has been formed now, it can directly enter into other steps of the glycolytic pathway. However, this dihydroxyestone phosphate which is being formed here, it cannot directly enter here. It has to get converted into glyceraldehyde 3-phosphate. That will happen in the next step, that we will observe in the next step. Dihydroxyestone phosphate and glyceraldehyde 3-phosphate, which are formed in the previous reaction by the breakage of fructose 1,6-bisphosphate. Now, these two components, out of these two components, glyceraldehyde 3-phosphate can directly enter into the further glycolytic reactions. However, dihydroxyestone phosphate cannot. Now, to overcome this step, we have this particular reaction where this dihydroxyestone phosphate will get converted into glyceraldehyde 3-phosphate. This, if you see here, there are these two components have the same molecular weight, they have similar molecular structure. Now, what happens is that this dihydroxyestone phosphate is a ketose, whereas this glyceraldehyde 3-phosphate is an aldose. If you see here, this carries CH2OH and then CO group. However, here we have the CHO aldehyde group, and this is a ketose group. Now, this dihydroxyestone phosphate will get converted into glyceraldehyde 3-phosphate by an enzyme called triose phosphate isomerase. Now, this reaction again, if you see here, the energy delta G for this reaction is 2.4 kilojoule per mole. That means it can be easily, reversible. It depends on the concentration of the products. What does that mean? That means that whatever the product has higher concentration can form the other substrates. Now, if there is a very high concentration of DHAP, then what will happen? DHAP will get converted into glyceraldehyde 3-phosphate. When the glyceraldehyde 3-phosphate concentration increases too much, then what will happen is that there will be formation of dihydroxyacetone phosphate. Now, this reaction is highly reversible. Now, what is the overview of this reaction? This is the overview of reaction. If you see here, there is a dihydroxyacetone phosphate getting converted into glyceraldehyde 3-phosphate. Now, if you see here, this ketonic group will get converted into aldehyde, and then here we see here CH2OH, hydroxyl group. Here CH2OH will get converted into CHO. Both of them carry 3 carbons. Only thing is that they differ here. In the ketone, here it carries hydroxyl group. Here it carries hydroxyl group, whereas it carries aldehyde group. Now, glyceraldehyde 3-phosphate, and then this is dihydroxyacetone phosphate. Now, this one in the reaction, what happens is that since glyceraldehyde 3-phosphate is continuously consumed by the next reaction, the concentration of glyceraldehyde 3-phosphate will become very, very low. Because of this, during glycolysis, dihydroxyacetone phosphate, the conversion of dihydroxyacetone phosphate into glyceraldehyde 3-phosphate is favored. Now, once there's a formation of glyceraldehyde 3-phosphate, the glyceraldehyde 3-phosphate gets converted into 1,3-bisphosphoglycerate by the action of enzyme called glyceraldehyde 3-phosphate, the glyceraldehyde 3-phosphate dehydrogenase. Now, dehydrogenases are the enzymes which remove anhydride groups from the molecules. So what happens here is that you can see here there is an hydrogen over here that will be taken out by this enzyme called glyceraldehyde 3-phosphate dehydrogenase. Now this glyceraldehyde 3-phosphate dehydrogenase uses an um, uses a coenzyme called NAD. Now, if you see here, this is the structure of NAD, where this is involved in various oxidation and reduction reactions. We will witness this even in case of citric acid cycle also, where these are very crucial enzymes. I mean coenzymes. This is NAD plus. This NAD, it is derived from vitamin B3 or niacin. You can see this niacin group over here. This is ribose and this is ADP. So what happens here is that this coenzyme takes up the protons and the electron to become here reduced into NAD plus H plus. Now this if you see here, this is a CHO group, that means aldehyde group here. This aldehyde will get oxidized to uh, carboxylic group over here. Now here what happens is that there will be conversion of this uh, aldehyde group into carboxylic group, and there is a formation of acyl phosphate group. Acyl phosphates are uh, the phosphates which are formed by addition of phosphate, the combination of carbonyl carbon and then with the phosphate group over here. This will form an acyl phosphate, and the other things are same here. If you see here, glyceraldehyde 3-phosphate. Now this aldehyde group will get oxidized to the carboxylic group, and then there will be an addition of a phosphate group here. And the difference, the distinction of this particular reaction is that the phosphate group over here, it is not derived from any of ATP or GTP or any nucleotide triphosphates. However, this will be from inorganic phosphate only. That is why there is no utilization of extra energy over here. Now what happens is that this glyceraldehyde 3-phosphate dehydrogenase take up this phosphate group. First what it will do, it will remove the hydrogen here, a hydrogen of this aldehyde, and it will convert that into carbonyl carbon, and then what it will do, it will add that inorganic phosphate. Now this particular group will become acyl phosphate group here. Now since it carries two phosphate group at carbon number 1 and carbon number 3, it is called 1,3-bisphosphoglycerate. Now this is glyceraldehyde because it has an aldehyde group over here. Now this is glycerate. Glycerate is a carboxylic group, carboxylic acid. Glyceric acid you can also call glyceric acid. However, it is in the physiological pH, it will be in the ionized form. Because of that, it is called glycerate. Now it is called 1,3-bisphosphoglycerate because on the carbon number 1 and then carbon number 3 we have two phosphate groups. Now this this is only the redox reaction that happens here. If you see all other reactions, they are all either phosphate transfer reactions or isomerization reactions or the aldol condensation reactions. However, only this is the reaction which belongs to a class of chemical catalyzed reactions called oxidation reduction, because your aldehyde will get converted into an carboxylic group. Now this also has a very the delta change between the reactants and the products is very low. Because of that, this reaction is highly reversible. The overview of this reaction I have written here: glyceraldehyde 3-phosphate plus PI. See, if you see here, this is not derived from any nucleotide triphosphate. However, it is derived from inorganic phosphate. I mean, it is freely available in the cytosol. That will be taken up by glyceraldehyde 3-phosphate dehydrogenase. And then this is another coenzyme here, NAD plus. Now this glyceraldehyde 3 aldehyde 3-phosphate will get oxidized to glycerate, and there will be addition of phosphate. Finally, we have this product 1,3-bisphosphoglycerate. Now, since NAD plus takes up the protons which are present on the aldehyde, it will get converted into NADH and then H plus. Now, as I told you, this is an oxidoreductase reaction, only reductase step, and it is highly reversible since uh, the reactants and products, the difference between the energy contents of the reactants and products is very less. This reaction is highly reversible. Now, 1,3-bisphosphoglycerate, which is formed in the previous reaction or previous step, in this step will get converted into 3-phosphoglycerate. Now it is just a transfer of phosphate group from 1,3-bisphosphoglycerate to ADP to form ATP. Now we have seen in the previous reactions, and usually what will happen is that ATP will give its gamma phosphate group to the substrates. However, in case of in this particular reaction, ADP will takes up the phosphate group from here, from the carbon number 1, and that will be converted into gamma phosphate group of this ATP. Now the enzyme for this particular reaction is phosphoglycerate kinase. Now this has been named kinase because the kinases are the ones which what will do, they will take the phosphate group from nucleotide triphosphates and add them to any other substrates. Now this is the overall reaction here. 1,3-bisphosphoglycerate, which is having 4 negative charges, plus ADP, which carries 3 negative charge, will get converted into 3-phosphoglycerate and ATP. Now in this step, this is a step where ADP, when ADP get phosphorylated to ATP, the phosphate group over here will be taken up by the ADP to form ATP here. Now when that goes, this there will be the remaining group is this carboxylic group. It will become COO minus. Now other uh, part of this molecule will remain same. Only thing is that this phosphate group here on the carbon number 1 will be taken up by the ADP to become ATP. And this particular reaction is catalyzed by phosphoglycerate kinase. Phosphoglycerate kinase is an enzyme which converts this 1,3-bisphosphoglycerate into 3-phosphoglycerate. Then again, this reaction is also highly reversible. This is highly reversible because the delta G value, the difference between delta G value of this substrate and the product is very low. Because of this less difference, now what happens is that this particular reaction will become highly reversible, and because of the energy difference is low, the reaction can flow in any direction. Either it can in the reverse direction, where there is a formation of 1,3-bisphosphoglycerate from 3-phosphoglycerate, or the conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate. Now both directions are feasible, but as I am continuously explaining the things, you can see that this reaction, the direction of these reactions is mainly guided by the concentration. Now if you have higher concentration of this 3-phosphoglycerate, then the forward reaction, I mean this reverse reaction is chosen. However, if you have more amount of this 1,3-bisphosphoglycerate, then this 3-phosphoglycerate, formation of 3-phosphoglycerate is preferred. Now to keep the structural structure of these molecules in check, now once you write this now what happens is that in case of this glyceraldehyde 3-phosphate, now the glyceraldehyde 3-phosphate, you have here you will have an aldehyde group, and then what will happen, this will become carboxylic group, and then again that carboxylic group will become the phosphate group on the carboxylic group will disappear. Now all these three reactions, 1,3-bisphosphoglycerate, and then formation of dihydroxyglycerolate 3-phosphate, and then formation of 1,3-bisphosphoglycerate, and then formation of 3-phosphoglycerate. If you see here, this particular part is same, but only the change will happen on the carbon number 1. In case of glycerolate 3-phosphate, you have this aldehyde group here without phosphate group, and then in this particular reaction where the glycerolate 3-phosphate get converted into 1,3-bisphosphoglycerate, the aldehyde will get converted into carboxylic group, and there will be addition of a phosphate group. And then in the next step, you can see here this phosphate group will be given to ADP, and again you will have a carboxylic group here. And this is very easy to remember if you properly write the structures. Now we understood how 3-phosphoglycerate will be formed here in the glycolytic pathway. Now this, in the next step, is that 3-phosphoglycerate, which is over here, now it will get converted into 2-phosphoglycerate. Now if you see the 3-phosphoglycerate and 2-phosphoglycerate, the structure of these two molecules, both of them are very same. I mean, they are same. Only thing is that the position of this phosphate group here, it is, in case of 3-phosphoglycerate, it is on the carbon number 3, because when we have this carboxylic group and this in a molecule, that will be given the carbon number as 1. This is carbon number 1, carbon number 2, and then carbon number 3. Now this phosphate group is situated on the carbon number 3 over here, and this phosphate, again it will come to the carbon number 2 over here to become 2-phosphoglycerate. And this reaction is very simple, and this reaction is catalyzed by an enzyme called phosphoglycerate mutase, 3-phosphoglycerate mutase. Or this mutases are the enzymes. What they do is that they just change the position of groups inside the molecule. Now a molecule is the one which is made up of many number of atoms and then groups. Now these mutases are the enzymes which change the position of these groups in the molecule. Now this phosphoglycerate mutase, what it will do is that it change the position of this phosphate from carbon number 3 to carbon number 2. Now to form, if you see here, this is 3-phosphoglycerate, which is having a phosphate group at carbon number 3, and then this phosphoglycerate mutase, what it will do is that it takes up that phosphate group, and then it will add that phosphate group onto the carbon number 2 to yield 2-phosphoglycerate. Now this is also a reversible reaction. Why I am continuously telling you that this here, if you see here, delta G value, this delta G value is very near to 0. That means it can be easily reversed. I mean, the enzyme can catalyze both forward and backward reaction, and that particular reaction depends on the concentration of the substrate or the products. Now this particular enzyme utilizes phosphohistidine intermediate. What does that mean? That means this enzyme as active site. In the active site, we have this histidine as one of the active amino acid. Now that phosphohistidine is an intermediate which takes place here during the conversion of this 3-phosphoglycerate to 2-phosphoglycerate. Okay, so this is the overview of this reaction where 3-phosphoglycerate getting converted into 2-phosphoglycerate. Now all the other things are same, only thing is that the position of phosphate group has been changed. This is a mutase type of reaction. As I told you, there is a phosphoryl shift from C3 to C2, and it is highly reversible. Now in this step, 2-phosphoglycerate, which is formed in the previous reaction, it will get converted into phosphoenol pyruvate. Now the difference between this and this compound is that the unsaturation, it is an unsaturated compound, whereas this one is unsaturated compound. Now that unsaturation is created by removal of a molecule of water. A molecule of water is removed by enzyme called enolase. Now this reaction is also highly reversible reaction. It can go to any of the direction. Now if you see here, the enolase is an enzyme which carries a magnesium ion in its active site. Now it catalyzes this particular reaction. Now during this reaction, what actually the enzyme do is that it takes out an hydrogen which is present on the carbon number 2 of this 2-phosphoglycerate, and then hydroxyl group which is attached to CH2, I mean carbon number 3 of this 2-phosphoglycerate, and they combine, and these two groups are combined and eliminated as a molecule of water. Now once water molecule is eliminated, or this OH and then H group is eliminated, you can see here when the hydrogen group is removed here, there will be a lone pair of electrons over here on the carbon atom, and then this OH group, if it is here again removed, again here there will be a lone pair of carbon electrons on the carbon. They will form bond here, which results in the formation of a double bond, C double bond CH2, and this particular compound, it is called phosphoenol pyruvate. Now this is the overall reaction here. 2-phosphoglycerate, it will get converted into phosphoenol pyruvate with the elimination of a molecule of water. It is a lyase reaction, lyase type of reaction, because it creates a double bond in the molecule with the removal of groups, that is water molecule, and again it involves the dehydration because there is an elimination of a water molecule, and it is highly reversible reaction. Now as I told you, there will be formation of 4 ATP here and 1 NADH here, and 2 ATP consumed here. This is the preparatory. This is called the conversion of glucose to fructose 1,6 bisphosphate. These stages are termed as preparatory phase, whereas this from glycerolated 3-phosphate to 1,3 bisphosphoglycerate and down this lane reactions are termed as payoff phase. In the preparatory phase, we utilize, and glycolysis utilize 2 molecules of ATP, whereas in the payoff phase it produces 4 ATP and 1 NADH. Now when we see the net output of ATP, so we have to minus 2 ATP which are utilized in the preparatory phase. Because of that, there will be formation of 2 ATP here. There will be only 2 ATP here and 1 NADH. Now recently, before it was thought that an NADH molecule can produce 3 ATP molecules, but recent literature and recent scientific experimentations have demonstrated that under anaerobic condition, an NADH, one molecule of NADH can produce approximately about 2.5 molecules of ATP. Because of that, there is a formation of 2 NADH molecules over here, and these 2 NADH molecules can give rise to each NADH 2.5. That means there will be formation of 5 ATP molecules. Now there are another 2 ATPs here, which will that will be remaining here. Totally it will be 7 ATPs which are formed in the glycolytic pathway. Now this is if you see here, in the glycolysis, in the glycolytic pathway, glucose is being broken down into 2 molecules of pyruvate with the elimination of so much amount of energy, and that energy will be trapped in the form of ATP only. Now though NADH which is being formed here in this particular step, it will be converted into ATP. The NADH in the electron transport chain will lead to formation of ATP only. Because of that, there will be total formation of 7 ATPs per molecule of glucose, which is getting converted into phosphoenol pyruvate. In the previous steps, we have understood how glucose will get converted into pyruvate, and also we have understood the energetics. What is the amount of energy which is being generated from the glycolysis in the form of ATP, that we have discussed. Now in this step, finally we will discuss on how this glycolysis is regulated. Now, as I told you, there are three important steps where the glycolysis is regulated. First one is hexokinase step. Second one is the phosphofructokinase. These are the three enzymes. And then another one is pyruvate kinase. The last step. The glycolysis is regulated by first step, last step, and the third step. Now, I had repeatedly mentioned that all these steps have very high energy difference, and all these enzymes are regulated by various components here. Now these are the main regulatory enzymes. These are the enzymes which get regulated so that the glycolysis can be regulated here. Now the first step is hexokinase. Now hexokinase is inhibited by glucose 6-phosphate. This is kind of a inhibition which we in biochemistry we call it as product inhibition, where the product of a particular reaction is only inhibiting that that particular enzyme or the particular reaction. Now this glucose 6-phosphate, if it increases in the cytosol, now that does mean that the glycolysis is not happening properly. That glycolysis is getting slowed down because there is already much amount of pyruvate or much amount of ATP, because glycolysis is part of the energy production, where the product, now once the glycolysis is completed, the product which is formed is pyruvate, and the pyruvate again enter into the citric acid cycle through the formation of acetyl coenzyme A. Again in the citric acid cycle, the acetyl coenzyme is completely oxidized. However, if the citric acid is getting slowed down, that does mean that there is slowed down electron transport chain. If there is an electron transport chain is slowed down, this is because of two main reasons. Either you have less amount of oxygen or you have more amount of energy. In both cases, citric acid cycle is inhibited. If citric acid cycle is inhibited, and then it will come down to the formation of pyruvate, and then it will come back to formation of glucose 6-phosphate. Now because of that, what will happen is that when there is a much amount of glucose 6-phosphate, that does mean that there is already the pathways are saturated and there is no need for the pathway to happen, or there is no need to the pathway to happen at that particular speed. Because of that, glucose 6-phosphate, again it will inhibit the hexokinase. This is the first regulatory step here, or the first regulatory enzyme here. And then next comes is phosphofructokinase. Phosphofructokinase is inhibited by one is ATP, citrate, and H+. Now it is obvious that ATP and citrate, or if you have more amount of ATP, what does that mean? That means the cell has been continuously producing more amount of ATP than it utilizes it. Now if you simply continue this catabolic pathway, there will be exhaustion of the product, and more amount of ATP, you cannot store very high amount of ATP in the cell, because you have to maintain a particular concentration of ATP. Beyond that you cannot go. So what the body will do is that body will store the excess of nutrients. Now those nutrients, whenever there is a requirement of energy, they can produce ATP. In that way, the ATP will inhibit this particular pathway, glycolysis pathway. If the glycolysis is inhibited, the glucose can move to the formation of glycogen and all. So in that way, you are reducing the unnecessary formation, the conversion of glucose into pyruvate, or unnecessary breakdown of glucose into pyruvate. Now that is why ATP is an inhibitor. Now citrate is the first the first component of citric acid cycle. Now this excess of citrate or excess of this H+ ions, now excess of citrate does mean that there is already much amount of citric acid cycle intermediates. Because of that, now if more amount of citrate means you have already saturated citric acid cycle. So because of that, again now this glycolysis becomes a feeder pathway for the citric acid cycle pathway. Now, so because of that citric acid, again, it indicate that the pathway should slow down. Because of that, ATP and citrate will inhibit the phosphofructokinase. Now, and then this particular enzyme is activated by AMP, ADP, and fructose 2,6 bisphosphate. That we will discuss that in another lecture, how this particular compound will be formed and how why this will activate this phosphofructokinase 1 alpha. Now, AMP, increase in AMP does mean that there is a high utilization of ATP. High utilization of ATP, that does mean that the energy, the cell is in a condition, or the physiological conditions are using more amount of energy in the system. If more amount of energy is utilized, you will have more amount of AMP. If you have more amount of ATP, that means energy is deprived. So in energy is less, the catabolic pathways are favored, and this glycolytic pathway is also a catabolic pathway, and this will increase if the AMP, ADP, and fructose 2,6 bisphosphate will go and bind or activate this phosphofructokinase 1, and that will result in formation of more amount of fructose 1,6 bisphosphate, and then further down the formation of pyruvate and acetyl coenzyme, and that can feed citric acid cycle, and then where you can get more amount of energy. And the last regulatory step is pyruvate kinase. This is the last step of glycolysis also. This is again inhibited by ATP, alanine, and cyclic AMP. This alanine is an amino acid which can be formed by addition of an amino group to the pyruvate. Now this will form alanine. Now if you have more amount of alanine, that alanine by transamination reaction can produce more amount of pyruvate. So because of that, what will happen here? If you have more amount of alanine, there is no need for this glycolysis to happen. Now this, if the glycolysis is inhibited, now that glucose 6-phosphate or glucose can move on to the formation of glycogen, or the storage. Glucose can be stored in the form of glycogen. That is why alanine, if there is an excess amount of alanine can inhibit pyruvate kinase. Now ATP, obviously for the same reason as here in this particular step, the excess of ATP can inhibit pyruvate kinase here. And then cyclic AMP, if you have more amount of cyclic AMP, it is an indication of there is a more amount of the energy in the cell, or there's an activation of pathways which indirectly indicate that there is a more amount of energy. That is why this cyclic AMP, alanine, and ATP can inhibit a pyruvate kinase. And again, this pyruvate kinase is activated by fructose 1,6 bisphosphate, and this particular mechanism, it is called feed forward reaction. Now what happens is that a product of a step can inhibit, can promote and or activate an enzyme which is down the pathway, activate an enzyme that is down the pathway, so that the pathway can effectively utilize the product which is being formed in this particular step. Okay, so now fructose 1,6 bisphosphate can activate this pyruvate kinase. Now this is about glycolysis. I tried to completely explain, and I have tried to simplify the reactions. I have tried to explain why that particular steps will happen, and how the regulation happen, and then what is the bioenergetics or energetics of this particular glycolytic pathway.